Hello, everybody. It's Dr. DeShavo. I hope you're doing great. And we are going to get ourselves into a little genetics here. I have a shortish little lecture on genetics because this is going to be an important part of your board. So we want to make sure we cover at least the basics. Of course, we could cover hours worth of genetics, but this is just a basic introduction into genetics itself. So let's take a look at that DNA. So, what is in that nucleus? General introduction. As we're looking at this picture, so just to sort of give you like a once over so you understand what we're talking about. We're going back to the cell. I know you guys love that. Okay, here's our little composite cell. A eukaryotic cell, don't worry about that. That just means that's one of our cells as opposed to a prokaryote. Don't worry about it. Just imagine this is one of our cells in general, just a composite cell to give you a general idea. Now, we know that we've got what's called cytosol, which is where all the junk that holds basically the organs of a cell. And inside of the nucleus, which is the brain of the cell, we are going to have DNA. Okay, and the cell, goes through different phases. Sometimes it's just resting and hanging out. Sometimes it's getting ready to replicate. It goes through different phases of what's called mitosis. And mitosis, you probably remember, is cell replication. Very good. Hopefully that just popped into your head. But when we're looking at the nucleus, if we have a cell that is just beginning to replicate, the genetic material that we know is usually in chromosomes, it's actually going to be like a big bowl of spaghetti. We're going to have these super amazingly long double alpha helixes of DNA, which look like spaghetti in a bowl. Okay, I'm not going to get into the whole specificities of what happens at this point, but realize that we've got these long strings of DNA. And as this nucleus approaches splitting during mitosis, because in mitosis we've got to replicate a cell and then split it, we will organize that long string of DNA into chromosomes. And chromosomes, we all know, are these X-shaped structures. And on these chromosomes, we'll have our genes. The genes are just the sequence of what are called nucleotides. And once again, I'm not going to get into that either. But located on these chromosomes, we have genes which will help to express the different characteristics of us as humans. Okay, and I did this little definition. I kind of stole some of this from the internet. Genes are the sequence of nucleotides, which are these base pairs. I don't know if you remember A, G, T, all those base pairs, okay, that are lined up here. I'm not going to get into it, but if it's ringing a bell, that's what we're talking about. So the sequence of nucleotides or nitrogenous bases in DNA in specific areas of the chromosome. Okay, the human genome has approximately 20,000 genes. Every time I go to teach this, honestly, I have to go back and really refocus on it because it's so complicated. Then I'm like, gee, why is it complicated? Because there's 20,000 genes. That's why it's complicated. So let's try to simplify this. We're going to talk about genetic material. The genetic material that we're talking about is found on this DNA, which is organized into that double alpha helix. It's a long string that is eventually condensed down into the shape of, we'll look at these chromosomes. There's several of them. And we find these chromosomes in the nucleus of the cell. On these chromosomes, guys, we have specific areas which will give us genetic information, okay? And the genes are located on those chromosomes. If that doesn't make sense, by all means, of course, shoot me an email. I'd be happy to clarify it more. So we can actually take one of those nuclei that have just begun the process of splitting, of replicating and splitting. And you could arrest it at an area or a phase called metaphase. I don't know if that rings a bell at all. It's one of the steps of mitosis. And geneticists or cellular biologists can arrest it at this phase and stop it and take the genes and actually put them into a karyotype. And you can see those genes are arranged according to size. And pretty much it goes from largest at 1, you could see, to smallest at 22. Although I believe that 21 is bigger, but they switch them for some reason. That is beyond me. And organize them according to size. And then they can organize them according to the genes as well. And we'll look at some examples of genetic mutations. And we'll say, I think one is at 15, one is at 22. We'll really focus a little bit more on the X and the Y. And we all know that the X and the Y are the sex-linked genes. You can see we have 1 through 22. We're going to have that general information about our entire body, excluding the sex-linked genes, which is your X and your Y for your males and your X and your X for female. What we're going to look at is this general, very basic introduction, but we're also going to look at changes in these genes, which can affect different parts of our body in many, many different ways. We're just looking at some very general examples. So the chromosomes, those X-shaped structures, let's talk specifically about those now that we've got a little bit of a picture in our head of what they look like. They're those X-shaped structures which contain genetic information. There's 23 pairs, which gives us 46 chromosomes. People get confused about this. So if you look back here, guys, there's 23 pairs, so there are 46 chromosomes. It's important that we have that specific number. If we have too few or too many, either we have genetic malformations or the fetus won't be viable. So just make note of that. People get confused about that. It's not 23 chromosomes. It's not 46 pairs. It's 23 pairs equaling 46 chromosomes. Okay? There's 22 pairs of those ones that I told you that affect everything except our sex chromosomes. Those are called autosomes, and 1 pair of sex chromosomes, which are XX or XY. Karyotype is that chromosome count that organizes them according to size, which I showed you, and also function. Okay. Phenotype is the observable characteristics from our genes. And we're going to go a little bit into Punnett squares. So for example, you may have a mother with brown eyes and a father with blue eyes. You'll do a Punnett square, which will take your genes and show you what phenotypes are probably going to be expressed. You know, are you going to have blue eyes? Are you going to have brown eyes? So phenotype is what you will observe. Now, patterns of inheritance and gene pairing. So it can be expressed as alleles, which are the variation of the expression of those genes, or those expression via the phenotype. Are you going to have brown eyes? Are you going to have blue eyes? Okay, so that's what an allele is. Now, homozygous means you have, say, the genes, suppose you have brown eyes and you only have homozygous for brown eyes, you'd have like a large B because it's a dominant and a large B. Heterozygous means you have different pairs of genes. So you may have a dominant gene for brown eyes, which would be a large B. I'm going to show this on a phenotype, on a Punnett square. I'm sorry, it'll be make a lot more sense. But then you might have a small b, which represents blue eyes. But because you have a dominant gene, we're going to get all into this. I'm getting ahead of myself. Okay, you're going to express that as brown eyes. Okay, so dominant will show up as a characteristic that's observable, and you will have at least one of those on the Punnett square. Recessive will be trump of the dominant if it's there, but if you have 2 recessive genes, you will express that recessive trait, such as blue eyes. Here, let's show you on a Punnett square, because this just makes it so much easier. So if we look at this Punnett square, we're looking at the expression of dominant and recessive. The father on a Punnett square, and this Punnett square you probably remember, will show the percentage of traits that may be expressed in a child. So this father and this mother are going to have a baby, and they would like to know whether that baby is going to have brown or blue eyes. The father has in his genetic makeup 2 recessive traits for blue eyes. He has blue eyes, so he is going to have 2 small b's, because in order for you to express blue eyes, you've got to have those recessive traits. The mother has brown eyes, so she could have a large B and a large B, because brown is dominant, okay? And that can be expressed either with a large B and a large B, or with a large B and a small b, because once again, dominant will always be expressed over recessive. In this case, the mother has a large B and a recessive, which is not being expressed, blue eye trait. They will pass these genes on to their child. This child has the possibility of having brown eyes, will be 50%. The reason being, in this Punnett square, this represents all their chances of their colors of eyes. So they have 25% chance in each square. Okay, oh, and you're supposed to, okay. So 50% will be brown eyes, and 50% could be blue. Why? Because dominant will always be expressed. So if they were to inherit this combination of their parents' genes, they will have brown eyes. If they inherit this combination of genes, they will have blue eyes. Okay? And we're going to look at 1 or 2 more examples of that. Of course, if you have any questions, shoot me an email. Okay. So, genetic and congenital disorders. So, things unfortunately can go wrong when babies are conceived and growing. There can be mutations that can affect their genes. There can be damage that can affect their genes. There could be nutritional deficiencies that can affect their genes. There are hundreds of disorders. Okay, and with the genetic that is passed down, we can characterize this in the Punnett square as well. So we can have a parent that carries it, which is dominant. We can have a parent which can carry a recessive, which will carry genetic disorders as well. So there's dominant genetic, deformations. There are recessive ones. So if a parent carries it, it depends on how those genes, once again, carry over from the mother and the father. If the child has a dominant genetic disorder and it's expressed the same way on this Punnett square, it would show up in 50% of the children. If it's a recessive genetic disorder, it will show up 50% of the time as well, if we make those B's either, dominant or recessive genetic disorders. So, okay, so we would use the genetic information from their parents. We would do the Punnett square, and we could determine if the dominant or the recessive genetic disorder can be carried over into the child. Okay. And I'm going to give you, I believe I have Punnett squares to give you an example of that. So we're going to go into dominant genetic disorders, recessive genetic disorders. So if you're only getting a little bit of this, wait until we get into those and we'll explain it even more. This is just a general introduction. Mutations, and once again, this is a general once over. We're going to go into the specifics in a slide or two. We could have single gene disorders in which a gene is defective on a chromosome. That allele or trait allele expressed could be dominant, it could be recessive. So dominant doesn't mean that it's always going to happen. It's that combination of the Punnett square gives us the chances of whether things are going to happen or not. So you've always got to look at that as well. This can happen in autosomes or those sex chromosomes. So you just have a genetic disorder on those 1 through 22 pairs that we looked at on the karyotype, those autosomes, or you could have a genetic disorder on the sex chromosomes. You could have a chromosomal disorder in the shape of the chromosome or structure of it, or the number of chromosomes, for example, trisomy 21. We're going to look at that. You can also have multifactorial disorders. There's issues with nutritional deficiencies, such as Spina Bifida we're going to look at. Smoking while you're pregnant, being exposed to carcinogens could also damage the genes that can cause genetic malformations in that child's genes, and it can be expressed. So with each of these general topics, we're going to look at specific disorders that can happen. I think it'll help to clear it up for you very nicely. So we're going to look at autosomal dominant disorders. These are dominant genes which will rule that Punnett square. If they're there, they're going to be expressed. The only way you're going to express a recessive one is if you have two recessive genes which come together. Some autosomal dominant disorders that we're going to look at are Marfan's and neurofibromatosis. You may be a little familiar with those. These will be transmitted from an affected parent and to the offspring. This has nothing to do with the sex genes themselves. So if we were to look at this on the Punnett square, you can see the affected parent is at the top. They have that dominant gene. That dominant gene will always be expressed with a large A. So bringing that large A down and down again, and then distributing the small a and the small a, okay, and then bringing the small a's over to the other boxes, we can see that there's a 50% chance that this will be spread. Now, this is just one example. Don't forget, with these Punnett squares, you might have two affected parents, right? And then you would bring those A's over, and you could see, of course, there's going to be a much higher chance of that happening and being transmitted. In this particular case, we will see 50% will get the trait. So let's look at some specific disorders. Marfan syndrome. This is a connective tissue issue, and this mutations on chromosome 15, and they can actually stain them, and they can look to see where that is on the chromosome itself. What happens with these patients or clients is they get very tall, they get very thin, they have long spider-like fingers, and they'll actually have it looks like a divot in the sternum, and they call it funnel chest. They can get scoliosis, and they can get heart defects. The aorta can be affected, and they can get damage to their valves as well. And it's all because they have a connective tissue disorder. They don't create and manage their connective tissue as well as they would without this genetic deformation. And they can have pretty bad heart problems, but you can monitor this because it will be obvious that they have it because they grow very tall, very thin, and I remember a friend of mine in college had this. He had really, really long fingers, and he moved very slowly because it was hard for him to control them. So with these patients, they will keep track of their heart to make sure that the valve defects don't cause a problem and too much stress on the heart. Neurofibromatosis, there's two forms. There's 1, 2, and there's actually a third type. We're going to talk about these two forms. And these are neurogenic tumors. Type 1 can affect nervous system the nervous system in general and cause tumors. And type 1 probably sounds a little familiar to you because they can get those cafe au lait spots. They can also get little bumps underneath their skin, subcutaneous lesions. They have a lot of freckles. And cafe au lait spots, I should explain that. Some people may have heard of this, some people may have not. They look, cafe au lait is when you put creamer in your coffee and you get that kind of creamy looking color to your coffee. They get these spots that have that presentation. They look like cafe au lait, coffee with a little creamer in it. Okay. They can get scoliosis, they can have erosive bone defects, but the big things that you'll notice is the lesions under the skin, the cafe au lait spots. I think the other really important thing to remember is the nervous system tumors, because they can damage their nerves and cause a decrease in nerve function in the area where they have those tumors. Type 2 is not as common as type 1, but the difficulty with this is it can affect the acoustic nerve, and they could affect other cranial nerves and spinal nerves as well. But the problem with damaging the acoustic nerve is you can lose hearing and or have balance issues, because don't forget the acoustic nerve is actually also vestibular cochlear nerve. It comes back to it, which affects balance and hearing. So there's really no treatment. There's palliative care, removal possibly of the tumors unless they are around a nerve, which obviously you can't damage nerves like that. So kind of a little bit of a sad disorder. Autosomal recessive disorders. So small but mighty. We're going to look at a Punnett square and see how that is put together. This is rare. It can affect both genders. 1 out of 4 will be affected. I'll show you that on the Punnett square. 2 out of 4 can be carriers. Onset usually will be early, and there'll be an early expression of these recessive disorders. Can present, in some cases, we're going to look as a deficient enzyme. Now, enzymes, don't forget, help to facilitate chemical reactions. So in some of these cases, like PKU, they don't have an enzyme that breaks down phenylalanine. So we'll look at that. Okay, so we're going to look at those 3 examples. First of all, let's look at that stinking Punnett square again. Okay, so if we look at this, we've got, with these carriers, it's recessive, don't forget. So we've got heterozygous parents, so they are both carriers. So what happens with the child is they have a 1 in 4 chance of being normal, a 50% chance of being a carrier, and a 1 in 4 or 25% chance of being affected with these autosomal recessive inheritance. Okay, so you would have to have a parent who, or 2 parents that are both heterozygous for this to have that outcome, because if this changed to homozygous for not having it, right, because it's a recessive trait, so you have to have the recessive gene, this would now become normal. This would now become a carrier, okay, and then this one would stay a carrier as well. So not to blow your mind, but in order to have an affected child, both parents would have to have the recessive trait. Okay? Just as a side note. So let's look at some examples. So cystic fibrosis is such a sad disease. It's autosomal recessive. It can be tested with DNA for diagnosis and also for carriers in a prenatal diagnosis. The expression of cystic fibrosis is an overproduction of mucus because they can't break that mucus down enough. Okay, and you could get lung congestion. But the other thing I always like to point out, and it's in the notes down here, so I want you to make a note of this, is not only do they have that lung congestion, they have malabsorption of nutrients because the ducts for the pancreas get blocked by mucus. And don't forget the pancreas, which we've got a little picture of it here, secretes digestive enzymes into your duodenum. we're going to talk about this in digestive. So the pancreas not only secretes and helps to, well, I shouldn't say secrete sugar, secretes insulin to regulate the balance of sugar into our cells from the bloodstream. It also releases digestive enzymes into your GI tract. And with cystic fibrosis, will block that duct of the pancreas, and they can't secrete digestive enzymes into the digestive tract. So make a little side note of that, please. So PKU is a mutation on chromosome 12. I guess I won't ask you the chromosomes these are on. I can't imagine they'd ask you on your boards. They might. I'm trying to give you like a general overall in some of these diseases. But anyway, I won't ask you chromosome number, but just so you know, these mutations happen on specific chromosomes. So they can't convert phenylalanine to tyrosine. Okay, both of these are amino acids. The problem with that is phenylalanine will then build up into your system, and it'll build up into the nerves of the body and the brain, and they cannot function properly intellectually. The great thing is we have early testing. We can determine it quickly, and this patient can have a diet low in phenylalanine, which you can find, I believe, in aspartame and meat products. Autosomal recessive disorders, Tay-Sachs. So this is progressive. Hexoaminidase is the substance that is the form that's necessary to break down certain lipids. If they can't break them down, these lipids accumulate and they will break down around the nerve cells, and the nerves get destroyed. And it leads to progressive mental and motor deterioration. Most are of Jewish descent, so they will do genetic testing to make sure that the patients don't have that. There's 3 forms: infantile, juvenile, and adult. Adult is really rare because you will generally find it early and hopefully treat it early, but I believe there's a lot of genetic testing for that. So these patients appear normal at birth. The infant begins to miss milestones. It can progress to seizures, muscle rigidity, any neurological signs or symptoms, and it can be fatal by 5 years of age. There's no cure. So once again, like I said, I believe there's a lot of genetic counseling to make sure that it's prevented. Sickle cell is also autosomal recessive. That's a defective hemoglobin, which doesn't create the right shape. So our hemoglobin and our red blood cells, okay, so our red blood cells, I should say, look like a Werther candy. So they're nice and round, and they are just the perfect size to fit right through a capillary, which is our smallest blood vessel. And the reason that's important is because we can offload oxygen and pick up carbon dioxide pretty easily, okay, because we have thin membranes there. But the other important thing about red blood cells being round is they can move through blood vessels very easily without getting stuck. With sickle cell, these patients, with their genetic predisposition for this, don't create nice round blood cells. They create a sickle-shaped cell. And a sickle is used by farmers to cut down hay. Those big ones like the Grim Reaper holds. I hate to make a weird analogy there, but so you can see with these sickle cells, they're not going to move easily through bloodstream. They're going to find other sickle cells, and they're going to lump together, and they're going to hook on each other, and they're going to get stuck in blood vessels that are tight, or generally in joints they like to get stuck, and these poor patients go into sickle cell crisis and have a lot of pain with this disorder. Okay, so another example, and I believe I talk about sickle cell a little bit more in the future, so I just want to go over it briefly here. So remember to make note of which is dominant, which is recessive, and know those disorders and some of their presentations. Sex-linked disorders. Is it the Y, or is it the X, or Y in general? Okay, they are generally almost always X-linked. And one of the examples we're going to give you is Fragile X syndrome. We're going to give you a couple actually. Okay, so Fragile X syndrome is associated obviously with the X chromosome. This can present as a long face, a large mandible. This is in males. Okay, large ears, large testicles, mental retardation, learning disabilities, speech delays, etc., etc. Okay, usually this will be recognized with a history and a physical exam that you could go furthermore and get genetic testing. There is not a cure for this. Hemophilia is due to an X-linked recessive trait, and this can cause, as you well know, excessive bleeding. There's different forms of this as well. We'll go into that when we talk about blood. Okay, there's other XY issues as well. Some of those actually will fall under the multifactorial inheritance disorders. So let's look at these. Multifactorial inheritance disorders, is it the genes? Is it environmental? Was it environmental that affects the genes? Somehow eventually the genes get damaged or affected, but there is probably some sort of environmental issue that has affected the genes, I mean. That's why your book has deemed it multifactorial, and I've actually taken this from another book, but anyway, so multifactorial inheritance disorders. So environment has affected the genes, extremely common, can be expressed at birth or later, and you could read through that list down there below. We're going to give you a couple of examples. And, you know, when it comes to these long lists in this lecture, realize I'm going to focus on the bigger stuff, okay? So cleft lip and palate is when, and this is probably very familiar to you, it could be the soft tissue in the front of the mouth, all the way back through the hard palate to the soft palate. This can come from smoking, diabetes, seizure medication, are all important risk factors in the mother. And this can be diagnosed in the ultrasound. Treatment is with surgery. They do such great surgery with this. Speech therapy. And then we want this fixed early, I'm sure, as you guys already know, because of suckling. Children who have too big of a cleft palate can't suckle, and that's bad for nutritional reasons for them. Spina Bifida, I put in there too, is when the neural tube doesn't close at the inferior end at the lower spine. It could be occulta, which is when we leave a small opening in the posterior of the vertebra, all the way to what is called cystica. They've stopped using these terms as much anymore. I'll show you the terms that they use in a minute. I've got a little quiz and answer practice section here. Cystica is when either the meninges and or the cord can come out of the posterior of the spine. And a little bit of an issue with this is that folate may be missing in the mother's diet. Okay, chromosomal number just may not add up. And yep, you probably already know the chromosomal disorder that we're going to talk about. So there can be an abnormality in chromosomal number and or duplication that happens in meiosis. And meiosis happens when sperm and eggs divide and get ready to join together so they don't have too many chromosomes, right? We can't have 46 chromosomes in an egg and 46 chromosomes in a sperm. That would give us way too many chromosomes. They actually will decrease down to 23 chromosomes so they can join together as 23 pairs of chromosomes, which is now 46 chromosomes. Did I just blow your mind? Okay, so in a normal cell, upon conception, we should have 46 chromosomes, 23 from the egg or oocyte, 23 from the sperm. Okay, we can get an abnormality in that chromosomal number, okay, such as Down syndrome. We're going to talk about that. There's more than 60 syndromes, so I'm just giving you one or two examples. Okay, Down syndrome is known as trisomy 21 because there is an extra chromosome at that 21 that we looked at on the karyotype. I'm going to show you a picture in just a minute. Increases with maternal age. You probably know most of this. Okay. The manifestations I'm not going to read through either because I think probably most of those look familiar to you. You can read through them on your own if you like. What I did find interesting, which I did not know, is that they can have congenital heart defects, most of these I didn't know. Ocular issues are more prone to leukemia, respiratory complications, GI complications, hypothyroidism. I never knew about that link. I also didn't know about the Alzheimer's link. Okay, so those two things were kind of new to me, as well as the ocular issues. So I actually knew about the ocular issues, but the Alzheimer's and the hypothyroidism I didn't know about. So parental screening, the 4D ultrasound, amniocentesis, hormonal levels are all ways to screen for that. Treatment, there is no treatment. We know it's symptomatic and supportive. And there's a picture of it right there, the extra one at 21. There's other disorders, which I don't think I get. Do I get into any more? Oh, I do get into one or two more here. Okay. Turner syndrome, which there's depletion of part or all of an X. This would be in females who have the XX. There's no Y chromosome implicated in this. So this would be a female. And you could see that they will have many manifestations, gonadal streaks instead of ovaries, which obviously you wouldn't be able to see that, but that lends themselves to being infertile. These patients are infertile. They tend to be short, increased weight. They have neck webbing, which means that their skin kind of webs out to the sides. They have a smaller lower jaw, drooping eyelids, small fingernails, widely spaced nipples. No mental retardation is present in these. So it's that physical manifestation of those characteristics that I listed for you. Okay, so Klinefelter syndrome has one or more extra X chromosomes with the presence of a Y. So this is a male. It's often undetected, but they tend to have more female characteristics. They tend to get gynecomastia, or they get small breasts. They have small testicles and a small penis. They tend to be tall. They have increased weight, not all the time, sparse body hair. May have learning disabilities, may have sexual dysfunction, varicose veins, osteoporosis, and breast cancer. Treatment is to give them testosterone, so they will have more male characteristics predominate. So with them, once again, there's a bunch of characteristics. I think the thing to remember with them is they tend to have more effeminate characteristics, such as breasts, smaller testes, smaller penis, sparse body hair. Okay, so, all right, so we are going to practice some of these. So, once again, as you're going through, go back. Try to remember what is dominant, what is recessive, what is linked to those sex chromosomes. Get a general working knowledge of the DNA, how that condenses into the chromosomes and organizes into the specific 23 pairs, that karyotype, and then specific characteristics. And, you know, I'm not going to get into like real nitty gritty, kind of like the big characteristics of each of these genetic disorders, just to give you an overview of what to remember from this. Because I know there's a long laundry list for some of these characteristics, so I want you to focus on the bigger things. So let's practice. I put together some pictures of some of these genetic characteristics. I would go back and maybe study a little bit before you review them. And as you're going through, really use this let's practice as a chance to sit and think about the physical characteristics of the genetic disorders that we just spoke about. Okay? So if you take a look at this, look at the mandible, look at the ears, think about those physical characteristics that we spoke about earlier. You can pause the tape if you like. Otherwise, we will take a look and let's see if you got it right. This is Fragile X syndrome. The larger mandible, the larger ears. I know this one is tough, so don't beat yourself up if you didn't get it. The other ones are quite a bit easier, but this was the only example that I had. Moving on. So, long arms, long legs. Really look at the chest. It's actually known as pectus excavatum, if you remember that, if you've ever taken a pathology course before. That depressed sternum. Marfan syndrome, you're right. And the fingers are long in this patient. Quite often, you know, they are very long and spider-like fingers, and they tend to move them very slowly. Yeah, this one's probably pretty easy. But which type is it? Neurofibromatosis type 1. So you've got those cafe au lait spots, you've got some of those subcutaneous tumors that are starting to present with this patient. Okay, so that one was much easier. And this one's very easy. Poor baby, though. You can see, I'm just going to pull down cleft lip and palate. So you can see the progression. This is a good example. You can see how it can progress really far back and can go all the way back to the soft palate where the uvula is with these poor little babies. But it doesn't have to. Some it's just the lip, some it starts to go into the hard palate, some it goes all the way back to the soft palate. And they do such great things, right, with the surgeries now. Thank goodness. Okay. And you know this one as well. Okay, Down syndrome. This one might be a little bit harder, okay? Think about what we have talked about so far. It's not the best example. We do have the drooping eyelids here. If we were to be able to see her neck, and this is a she, maybe she'd have a little webbed necking, okay? I may have given you some clues. Once again, it's not the best picture in the world, but this would be that Turner syndrome, okay? This one's probably a little bit easier, okay? I should have given you a heads up. Never study when your kids are around in this class because you never know what's going to show up. Think about what we've spoken about so far. Small penis, small testicles, slight gynecomastia on this patient. And this is Klinefelter's, which we spoke about before. Now, the last one, okay? Yeah, you got it. It's Spina Bifida, but these are the different forms. And some of you may be more familiar with this than others. So this is what I used to call occulta, okay? And it was called occulta. And notice they don't call it that anymore. I'll show you what they call it in just a minute. And there was the lack of the spinal vertebra in the back. I'll erase that in just a second. And most of the time, they're going to have hair, a little patch of hair over that area. And let me erase that. So these patients have that opening here where the back part of the vertebra, so this is the front part, and don't forget, this vertebra actually is like a ring all the way around that protects the spinal cord. In these patients, that ring is not complete. It's open. So the spinal cord is here. It's safe. It's covered by fat and muscles and connective tissue. But these patients still have an incomplete circle of the vertebra in the back. So this is Spina Bifida occulta. Now, these two forms that are left over, I had learned at Cystica. They've broken them down into two types. And the first type is the one in the middle, which is called meningocele, which is when the meninges only come out of that opening, and you can see are exposed on that posterior aspect of that baby's back. And the next step is myelomeningocele. What that means is the spinal nerves. Let me draw for you. So this is about L2 because it's the end of the spinal cord. So with myelomeningocele, the meninges as well as the nerves of the spine and or the spinal cord can go back into that posterior pocket, which can be exposed. Now, these two are obviously surgical interventions that needs to be fixed. This people can live with for years without a problem. Okay, so Spina Bifida. And that, my friends, brings me to the end of this genetics lecture. I hope this makes sense. Genetics is so tough to teach, but as always and forever, if you have any questions about this, shoot me an email. I'll be happy to get back to you. Have a great day.